ELAB is a fantastic data acquisition system for recording single-unit action potentials, local field potentials, long-term potentiation, EEG and ECG signals. ELAB has 50K sample per second sampling rate per channel and is the only electrophysiology device that covers all extracellular techniques. For more information about ScienceBeam products, please visit our website.
Hello everybody, I hope everyone are fine during this pandemic and new peak of the disease. I'm so glad to meet you in this presentation. Previous I present my lecture for my students and it was not easy, but now all of you are excellent professors and it got very difficult for me to lecture for you. Anyway, I try to do my best, and if you let me, I can start. Sorry, if you turn off your sharing, after that I can share my slide.
Sure.
Sorry, can you see my slide and can you hear me?
Yes, doctor, I have your voice and also
yes,
your...
Okay. Very good. Thank you for attending my presentation. I want to explain about synaptic plasticity, the cellular mechanism of learning and memory. My name is Farham Raisi, and in this lecture I want to speak about some introductory about learning and memory. After that I will explain about field potential recording and some basic aspects of its same to current things and sources. Also, you need to know about excitatory postsynaptic potential when you are recording from extracellular fluid. After that, I will continue with long-term potentiation, LTP, long-term depression, LTD, paired pulse facilitation, PPF, and paired pulse inhibition or depression, PPI or PPD, that are different forms of synaptic plasticity in hippocampus. Also, you can record this synaptic plasticity in other places, but in other regions of the brain, but I want to explain about them in hippocampus. First, about learning and memory. Sorry, I know that you know everything about this introduction, but I have to remind them. After that, I will continue with the cellular mechanism of LTP. As you know, our power to change our behavior according to our experience is learning, and memory is the final from learning that is essential for saving this learned experience. As you know, every behavior, every activity from us need to memory. Without memory, we have to do everything same to first time. Because in the first time, by doing some try and error, we learn about the best activity. After that, we save this experience and we can do our best in future. And without this memory, we learn about the best activity. After that, we save this experience and we can do our best in future. And without this memory, we have to do this try and error again and again. And this is very difficult, and therefore memory is essential for our survival. And we have to know everything about this for prevention of memory loss and of also about treatment of disease same to Alzheimer's disease, same to diabetes or other diseases can impair learning and memory. But if I want to explain the mechanism of memory, memory in fact is convenience of passage of signal through synapse. Consider a field that is covered with snow. When you want to walk along this field, you have a lot of difficulty during the first walking. But in second time, because you can put your feet and your legs on footprint of previous walking, you have less difficulty. In the third time, fourth time, or when you repeat this action many times, by time you can produce a road and eventually you can pass very easy through this field. Also, we have this situation in synapse. When for the first time you read a book, you have a lot of difficulty to understand it. But if you read it many times, after that, when you see that reading, instantly you remember everything. And the key element of memory is repetition. When you repeat this action a few times, you can save this memory, this experience for a short period of time. But if you repeat it many times, therefore you can keep it for a long time and sometimes for your lifespan. In addition of repetition, another factor that is very important is motivation. Sometimes when I was a student, when my professor explained about learning and memory, I couldn't understand because I didn't have enough motivation in that time. I studied before my exam. But now I have to explain for my student, and therefore I have to know everything. I have more motivation, and when you have more motivation, when you study anything, you can memorize it very fast. Therefore, motivation and repetition are very, very important for memory. We have two types of memory, short-term memory and long-term memory. Short-term memory has two types of memory, sensory memory and primary memory. In sensory memory, consider, sorry, can you see my whiteboard?
Yes.
Sorry, because I want to switch to paint and my slides many times. Please, if you have any difficulty, please inform me. Thank you. Consider we have presynaptic neuron and postsynaptic neuron. If you stimulate one neuron one time, because of release of neurotransmitter in synaptic cleft, we have one action potential in postsynaptic neuron. But until the time that this neurotransmitter is present in synaptic cleft, we have repeated action potential in postsynaptic neuron. And after milliseconds to seconds, by removing of this neurotransmitter, by degrading or escaping from this space, we have the end of this action potential. This can remain, as I said, for milliseconds. For example, if you see your light in your room, after that, close your eyes. After a millisecond, until the millisecond, you can see light in your eye, but it will disappear by time. This is
this is sensory memory. But primary memory, in primary memory, we have input neuron and some circuits, reverberatory circuits. One action potential comes to these reverberatory circuits and can circulate in this reverberatory circuit, and every time reach to output neuron produce one action potential. Therefore, one input, we have a lot of number of output. An example about this type of memory, if you ask a phone number until the time that you repeat that number, you can remember it. If anybody asks, what are you doing? Exactly, you forget. Therefore, this type also is a short-term memory. But in long-term memory, we have two types of memory, secondary memory and tertiary memory. In secondary memory, you can save this memory for days to months, but eventually you will forget this type of memory. But in tertiary memory, because of some structural change in synapse, you can keep this type of memory for a long time and your lifespan. And for secondary memory, we have some chemical changes in synapse, and also some transient activity same to early LTP. But in tertiary memory, we have a structural change and also late LTP. By this situation, we can keep this information for a long time. Long-term memory has two forms: implicit memory or non-declarative, that you can't explain about this memory for other person because they are skills, your emotion, and also your reflex. But explicit memory or declarative memory, you can explain about them because they are facts or events. For example, you went to restaurant yesterday with your friend, or apple fall down because of gravity. In this presentation, I want to explain about explicit memory and its cellular mechanism, synaptic plasticity. You have to know about the regions of the brain that are important for explicit memory. First, prefrontal cortex, that is important for working memory. And after that, hippocampus. Hippocampus is a place that when say to your brain, this information is important, after that, it can convert it to long-term memory and you can save it for a long time. And the cerebral cortex is the ultimate storage site for memory. When hippocampus convert any memory to long-term memory, you can save it in cerebral cortex for a long time. But because I want to explain about the mechanism, synaptic plasticity mechanism in hippocampus, for better understanding, we have to know about the hippocampus, structure of hippocampus, circuits, neural circuits, and also its connection with cortex. The main input of hippocampus originated from entorhinal cortex. Entorhinal cortex is part of medial temporal cortex that is responsible for processing of multimodal sensory and spatial information. And the main output of hippocampus originates from CA1 region. CA1 has pyramidal neurons and is the main output of hippocampus. The main input of hippocampus that originate from entorhinal cortex reach to hippocampus through perforant pathway. And we have two direct and indirect pathway to hippocampus. Also, we name the indirect pathway trisynaptic pathway. In indirect pathway or trisynaptic pathway that originate from entorhinal cortex layer 2, then it's in projection to neuronal cell in dentate gyrus of hippocampus and stimulate the granule cells in this area. After that, axon from granule cells innervate CA3 area, and the CA3 area has pyramidal neurons. The pathway that sends signal from dentate gyrus to CA3 is Mossy fiber pathway. From CA3, we have axons that project to CA1 area and innervate pyramidal neuron in this area through Schaffer collateral pathway. But in indirect pathway from perforant pathway that originate from entorhinal cortex layer 3, innervate CA3 pyramidal neurons or CA1 pyramidal neurons and CA3 pyramidal neurons directly. Therefore, we have direct connection to CA1 or CA3, or indirect through dentate gyrus, CA3, and after that, CA1. In addition to this, we have some interconnection between neurons in CA3 area that they can self-stimulate, and this is and excite themselves. Therefore, CA3 has high excitability, and sometimes in pathological condition are center for induction of seizure, because hippocampus, in spite of cortex that has 6 layers, hippocampus has 3 layers, and because it has high excitability and because of interconnection of CA3 area, is a good place for having kindling model, and through electrical stimulation you can induce and kindle rats for evaluation and research in context of epilepsy and seizure. In addition to this pathway, also we have another region in hippocampus, CA2 area. CA2 area located between CA3 and CA1. CA2 receive input from entorhinal cortex from direct and indirect pathway, also receive a strong input from hypothalamic nuclei that release oxytocin and vasopressin. The hormones that important for our social behaviors. Therefore, in future, if you want to research about social behavior, it's better to stimulate, for example, perforant path and record from CA1, or directly stimulate CA2 area, after that record from CA1 area. By this way, you can use this area for different aims. Anyway, CA2 sends a strong output to CA1 area. Therefore, we have direct, indirect, and also from CA2 area to CA1 region of hippocampus. After that, from CA1, we have projection to different part of cortex for storing of our memory. Now I want to continue with field potential recording. First, I want to explain some introduction. After that, I will continue with synaptic plasticity. We have 2 types of recording: intracellular recording and extracellular recording in electrophysiology. If you put one electrode inside the cell as recording electrode and another, for example, at the level of skin as reference electrode, and you can measure the voltage difference between these 2 electrodes. And because according to agreement by electrophysiologists, we consider the voltage of extracellular fluid equal to 0, and we attribute all of the voltage difference between recording and reference electrode to intracellular space. And the form of action potential, the stimulation, EPSP, threshold level, depolarization, repolarization, hyperpolarization, and polarized level. This form of action potential is result from intracellular recording. But field potential recording is result from extracellular recording. In extracellular recording, we put both electrodes outside of the cell. one at the level of skin as reference electrode, another near to the membrane of cell as recording electrode. As you know, because inside the cell is negative and outside of the cell is positive, and as I said, we consider the voltage of extracellular fluid equal to 0. When this cell is silent, you can record 0 voltage between these because both of them in extracellular fluid, and therefore you can record 0. No voltage difference between these two electrodes. But when you stimulate this neuron, this positive ion come into the cell and make inside the cell positive, but previously this positive charge located beside one negative charge. When this positive come into the cell, this negative charge remain uncharged and make near the cell negative, and this electrode record a negative deflection in voltage. Or sometimes you inhibit a neuron, for example, by release of by release of GABA. We have opening of channel, chlorine come into the cell, or potassium exit from the cell, therefore make inside the cell negative, more negative, therefore extracellular fluid get more positive. During this situation, you record positive deflection in your recording. This one is 0, and this one can detect any voltage difference in the cell, but opposite in extracellular fluid. Now, therefore, in this presentation, I want to explain about field potential recording, that is type of extracellular recording. In my previous lectures, I explained that in extracellular recording, the key element and the main criterion of recording is the size of your electrode. When you use very tipped electrode, because this electrode has high resistance, therefore you can pass only a strong signal that are near to this electrode, and this method is suitable for a single unit recording. But if you use electrode with thicker size, because you have a lower amount of resistance, therefore you can gather electrical activity of different cells near to this electrode. Rather than one action potential, we can gather the consequence of electrical activity of different cells. And in multi-unit recording, because our size is higher than this type for single, but is not very high, therefore you can record only from a few number of cells in multi-unit recording. But if you increase the size of electrode, lower resistance, therefore you can record from large population of cells, a field of cells. For example, from a nucleus in the brain. And because of activity of different cells add to each other, and this consequence can record by this electrode. And for LTP, LTD, and PayPal facilitation or depression, we use this model of recording, field potential recording. And if you very thick electrode, you can record from whole tissue. For example, in electrocardiogram or electroencephalogram, you can record from a very large field of cells. Before explaining about field potential recording, you have to know about excitatory postsynaptic potential. When we have two neurons, presynaptic neuron and postsynaptic neuron, if you stimulate this one and this neuron release excitatory neurotransmitter same to glutamate, because of opening of, for example, sodium channel, we have inside, when you are recording from inside the cell, your electrode is located inside the cell, because by opening of sodium channel, sodium come into the cell and make inside the cell positive. we name this voltage change excitatory postsynaptic potential. And if you are recording from intracellular, therefore you record positive deflection. But when you are recording from extracellular fluid, because when inside the cell get negative, outside of the cell get, inside the cell get positive, outside of the cell get negative. Therefore, in extracellular recording, when you have EPSP or excitatory postsynaptic potential, you have negative deflection. If you release GABA neurotransmitter and you are recording from intracellular fluid, you record negative deflection. But if you are recording from extracellular fluid, you have positive deflection. We are not concerned about IPSP and hyperpolarization. I want to explain only about EPSP and excitatory postsynaptic potential. Because you are recording from extracellular fluid, therefore, we are concerned about only this one. You have negative deflection. EPSP in extracellular recording is negative deflection. But now I want to explain, we have sometimes EPSP, but positive deflection. Sometimes negative, sometimes positive. Why? Let me to explain about dipole. Consider we have two negative and positive poles. Between these two poles, we have electrical field. If you locate your electrode near to positive pole, you can record positive deflection, this area, negative deflection. We have a mechanism named current sinks and sources. When you have a cell axon, inside the cell is negative, outside of the cell is positive. And because we have infinite number of positive and negative ion in extracellular fluid, we consider the voltage in extracellular fluid 0. And we put our electrode near to cell membrane. When you stimulate a neuron, and by opening up some ion channel, this positive charge come into the cell and make this part of the cell positive. This part is negative, this part is positive. Also, this part of extracellular fluid get negative, because previously we had both negative and positive. Now we have only negative charge, more negative charge here. We have dipoles between this area and also this area. And because of leak channel of potassium and opening of sodium channels, we have complete circuit of electrical current between these two areas. Therefore, when you stimulate a neuron, by opening of sodium channels, sodium come into the cell, make inside the cell positive. And in extracellular fluid, in this area, we have negative charge. We name this area current sink, that vacuum the current toward the cell. If you put your electrode in this part of the synapse, you can record negative deflection, as I explained in previous. But because of this dipole, we have a current of positive charge toward negative charge, and this positive charge, because of high concentration, exit from the cell, and also we have positive charge toward negative charge. We have accumulation of positive charge here. Now, if you put your electrode near to SUMO in current source, you can record positive deflection. Therefore, you can see in field potential recording and in extracellular recording. In both situations, you have EPSP, but according to the place of and location of your recording electrode, sometimes you record this EPSP as negative deflection, and if you put far from synapse and near to soma cell body, you can record positive deflection. Therefore, I will show you when you are recording in CA1E area, you have negative deflection because you are recording from dendrites and far from soma. And in dentate gyrus, we usually drive down toward soma and cell bodies of neurons, and we can record a positive deflection in dentate gyrus. Also, you can see this part is sink, this part is source. If you put your electrode near to sink, you have negative deflection, and near to source, you have positive deflection. Both of them are EPSP. This EPSP is not result from activity of only one neuron, rather than, as I said, because we have low resistance of our electrode, it can gather electrical activity of a group of cells, and this is a consequence of the activity of a group of cells, and we have this negative deflection in CA1 area. This is a real recording from my research in my lab, and this one also is result from my thesis, that positive deflection in dentate gyrus. This EPSP is result from consequence of electrical activity of a population of cells. But in addition to EPSP, we have this indentate on EPSP. We name it population spike, or PS, indentate gyrus. What's the reason of this indentation? Because when you stimulate a group of cells, some of them, because of high excitability, reach to threshold level and can produce action potential. During action potential, all part of the cell, inside the cell, get positive, and all part near to extracellular fluid, near to neuron, get negative. Therefore, in consequence of a group of neurons, we have EPSP, because a small number of neurons produce action potential, make extracellular fluid negative, and produce this indentation. And we name it population spike, or a group of action potential that accompany with this consequent activity of different neurons. Long-term potentiation, that was firstly explained by Bellis and Lomo in 1973. In hippocampus, we have different types of LTP, and in different types, different receptors, ion channels, and also second messengers and metabotropic receptors are involved. In some of them, we have change in presynaptic neuron, in some of them in postsynaptic neuron, in some of them in both of them. In relation with LTP, you have to know about Hebb's rule, Hebbian theory, that was firstly explained by Donald Hebb. He said, when an axon of cell A excites cell B repeatedly and persistently, play a role in activity or firing of the B neuron. Because of some structural and biochemical changes that can occur in one or both of these cells. And eventually, this process, these structural and biochemical changes can increase the synaptic efficacy. You have to consider in Hebb rule, we have to have simultaneous activity of pre and postsynaptic neuron. But presynaptic must activate just before postsynaptic activity and firing. According to Hebb's rule, if we have change in postsynaptic neuron or both pre and postsynaptic neuron, we have associative LTP. But if we have only change in presynaptic neuron, we have non-associative. Maybe in textbook you have encountered with associative and non-associative. Therefore, non-associative, we have change only in presynaptic neuron, but in associative, we have change in both postsynaptic, only postsynaptic or both postsynaptic and presynaptic. Anyway, Hebb's rule simply say that cells that fire together, wire together. In hippocampus, the main neurotransmitter that is important for induction of LTP is glutamate. Glutamate, as you know, has different ionotropic and metabotropic receptors. Two ionotropic receptors are very important. First one is AMPA receptor that is permeable to sodium and potassium, especially to sodium, because we are near to equilibrium voltage of potassium. However, it's permeable to potassium, but we don't have any driving force, and we have more passage of sodium and make inside the cell positive and produce depolarization. And another receptor, ionotropic receptor, is NMDA receptor that is permeable to calcium. For opening of AMPA receptor, presence of glutamate is enough. But for NMDA receptor, in addition to presence of glutamate, also we have to stimulate the postsynaptic neuron and depolarize that and make depolarized that neuron. After that, we have opening of NMDA receptor. Why? Because magnesium block the entrance of an NMDA receptor from extracellular side. Magnesium is bind to NMDA receptor and block the entrance of NMDA receptor and prevent from opening of it. Therefore, we need to depolarize the postsynaptic neuron by this depolarization and structural change in the NMDA receptor. After that, we have removing of this magnesium and that NMDA relief from that magnesium. And by presence of glutamate, we can open this channel and calcium can come into the cell. As I said, in different part of hippocampus, we have different types of LTP, associative and non-associative. First, I want to give you some example about associative can occur in synapses between perforan pathway and also granule cells in dentate gyrus. And also, we have associative LTP in synapse between Schaffer collateral pathway and pyramidal neuron in CA1 area. If you put your stimulating electrode on Schaffer collateral pathway and record from pyramidal neurons in CA1 area, after first you have some basic response. After that, using high frequency stimulation. I have to say, I said, if you want to learn, you have to repeat. In LTP, we don't have time to repeat and study one book many times during different days. We want to induce this memory instantly. And for this, in lab, we have to, for induction of LTP, we have to apply high frequency stimulation or tetanic stimulation. After tetanic stimulation, for example, a burst of 100 hertz, we have induction of LTP. Let me to show. You have presynaptic neuron, postsynaptic neuron. If you stimulate the presynaptic neuron with this intensity, you have EPSP with this size. If you apply high frequency stimulation, after this hypertetanization and tetanic stimulation, if you apply same intensity to previous, you can record very high amplitude EPSP in postsynaptic neuron. This is LTP. Simply, this is LTP. Because of some structural or chemical change in presynaptic and postsynaptic neuron. Before tetanization, response to this intensity of stimulation is very small. After that is very large. After tetanization, we have induce of LTP and induction of LTP, and we can maintain it for a long time. But if you apply AVP, that is antagonist for NMDA receptor, by blocking of NMDA receptor, as you can see, they prevented from LTP induction and its maintenance. And this show you that this type of LTP is depend on postsynaptic neuron and depend on NMDA receptor. This type of LTP is associative because it depends on postsynaptic neuron. But in synapse between mossy fiber pathway and pyramidal cells in CA3, we have non-associative form of LTP. If you stimulate, you insert your stimulating electrode to hippocampus and stimulate mossy fiber and record from pyramidal cells in CA3. After first we have, you can see we have basic response after tetanization and applying high frequency stimulation. In green bullet, we have induction and maintenance of LTP. If you apply APV blocker of NMDA receptor, you can see we don't have any change in LTP induction and maintenance. That show you in this site, we don't need to NMDA receptor and postsynaptic neuron. But if you apply protein kinase A inhibitor, you can inhibit significantly the LTP induction and maintenance. In fact, in mossy fiber, after high tetanic stimulation, we have increase in calcium concentration in axon terminal. Sorry, axon terminal. This calcium, by activating protein kinase A, and protein kinase A by phosphorylating different proteins, increase the trafficking of vesicles and release of neurotransmitter. Therefore, after tetanization, we have a change that by each stimulation, we have more release of glutamate, more concentration of glutamate, we have larger EPSP. After tetanization, because of change in presynaptic neuron, we have higher concentration of glutamate. Therefore, this higher concentration can stimulate more the postsynaptic receptors, and we have more amplitude EPSP in postsynaptic neuron. This type is non-associative, and if you inhibit protein kinase A, you can prevent from release of this neurotransmitter, and you can suppress the induction of LTP. In future, you need to know about induction of LTP and expression of LTP. In induction, we have biochemical, in fact, induction of LTP is the biochemical reactions that activate by high frequency stimulation or tetanic stimulation. 1 burst, 4 bursts, or for example, in CA1, I usually use 4 bursts of 100 hertz of stimulation. In dentate gyrus, I use 10 bursts of 400 hertz of stimulation. We have different protocol according to your aims. But expression is the long-term change that is responsible for enhancing in synaptic transmission and efficacy. In my explanation about memory, I said that in long-term memory, we have secondary memory and tertiary memory. Because we have early phase, early like in LTP and late LTP. In early LTP, if you apply only one train, one burst of stimulation, one burst of 100 hertz stimulation, you can induce LTP, but you can maintain it for a short period of time, 2 or 3 hours. After that, it declines and eventually disappears. Therefore, it's suitable for short-term form of memory. But I said, if you pass one time through that field that covered with snow, in first time you have some difficulty. But when you apply a train of stimulation, after this repetition, you have less difficulty. But in late LTP, if in addition you have 10 people that are your friends and all of you pass through this field many times, therefore you can make a street and easily you can pass through that field. If you use rather than one burst of stimulation, you apply 4, 3, 10 or more 10 bursts of stimulation, you can see we have gradually increase in induction and responsivity of that synapse, and also we can maintain it for days, many days. Because in early LTP we have only a chemical change, but in late LTP we have, you can see let me explain first about this slide after that. In early LTP, we have chemical change, I will explain. And in late LTP, we have a structural change and expression of new proteins and new structure in the synapse. Now I want to explain about mechanisms that can occur during LTP. Sorry, the organizer of the program, if you prefer to give some minutes for rest, I can give you this time. If not, I can continue. Okay, to save our time, I continue. First, I want to explain about the synapse in hippocampus. We have presynaptic neuron and postsynaptic neuron. I said the main neurotransmitter in hippocampus is glutamate. After action potential in presynaptic neuron and release of glutamate, we have glutamate in synaptic cleft. We have two main receptors, ionotropic receptors of glutamate, AMPA receptors and NMDA receptors. NMDA receptors, because of presence of magnesium, are blocked from extracellular is blocked by magnesium from extracellular side. Because of presence of glutamate, this glutamate can bind to AMPA receptor and open it. After this opening, sodium comes into the cell and will depolarize the intercellular space of postsynaptic neuron. This depolarization usually is not enough to remove this magnesium. Therefore, we don't have any opening of this channel and coming of calcium. We don't have any LTP. But if you train a burst of stimulation, because of this burst, we have released high concentration of glutamate. Therefore, we have summation of this stimulation, and we have adding of different depolarization result from AMPA receptor, eventually reach to a threshold level, and we have action potential. This depolarization, this enough depolarization can remove this magnesium, and because we have glutamate, this glutamate can open NMDA receptor, and therefore calcium come into the cell. Now this calcium start many different actions inside the cell. Calcium can bind to calmodulin, and the complex of calcium calmodulin can activate different enzymes. One of them is calcium calmodulin protein dependent kinase 2 or CAM kinase 2. This enzyme can phosphorylate AMPA receptor. By this phosphorylation, the sensitivity of AMPA receptor will increase. Also, it gets more activated, and therefore it can pass more number of sodium and better function for more depolarization. And by having higher amount of depolarization, we can remove this magnesium, and the final result is higher concentration of calcium. This is very helpful in induction of LTP. Also, this calcium calmodulin complex can activate protein kinase C. This protein kinase C that is dependent on calcium calmodulin complex can phosphorylate some AMPA receptor that are made in previous, and they stored in some vesicle. By phosphorylation of this AMPA receptor, they move toward to dendritic spine and insert this AMPA receptor to this synaptic place, and therefore we have more number of AMPA receptor, more depolarization. The final result is more calcium. And more calcium, as I explained in previous, we have LTP induction. And also this calcium calmodulin complex, by activating nitric oxide synthase, increase the synthesis of nitric oxide, a gas that acts as a neurotransmitter, and also in display act as retrograde signal that release from presynaptic neuron and release from postsynaptic neuron, and can reach to axon terminal, and by affecting different proteins in this place, increase the trafficking of vesicles and release of glutamate. By this way, we have increase and strengthening of synaptic transmission. All of these mechanisms are important for early LTP, and we can keep this situation only for 2 or 3 hours because of one train or one burst of stimulation. But in late LTP, if you apply in CA1 4 bursts of or 4 train of stimulation, or in dentate gyrus 10 bursts of stimulation, because of this high frequent stimulation and tetanization, we have higher concentration of glutamate, and because of this situation, we have more concentration of calcium. When we have very high concentration of calcium, we have a starting of late LTP. Late LTP through 2 functions can induce LTP and promote these long-term changes. One of them is this part. We have an enzyme named protein kinase M-zeta. This protein kinase M-zeta is same to protein kinase C, but protein kinase C is dependent on calcium calmodulin complex, and activation of it needs to calcium, presence of calcium. But this one is independent from calcium and don't need to calcium, and constitutively is active and can phosphorylate this AMPA receptor that is stored in vesicle and therefore move them to spine, dendritic spine, and increase the responsivity of presynaptic neuron. But this enzyme needs to be made. We have some prefab and made mRNA that are present in previously. We have transcription of this mRNA. These mRNA are present in cytoplasm, and by very high concentration of calcium, we have increase in translation of this mRNA and making protein kinase M zeta, and after that it can continue phosphorylation of AMPA receptor without presence of calcium. It's an effective way for late LTP. Also, during late LTP, we have recruitment of adenyl cyclase. That by activating of this enzyme, we have increase in concentration of cyclic AMP. After that, it activates protein kinase A, and by affecting cyclic AMP response element binding protein, increase transcription from genes, and after translation, we have new effectors. These new effectors are NMDA receptor, AMPA receptors, also some proteins that are important for making a new dendritic spine, and also by making some retrograde signal increase the interaction between presynaptic and postsynaptic neurons. Because of this structural change and expression of protein, we have long-term memory, and it can present for days, years, and sometimes our lifespan. You can see in early LTP, if you block and inhibit protein synthesis, you don't have any change in LTP induction and maintenance because we have some chemical change. But in late LTP, if you block protein synthesis, we don't have any problem in induction, because induction needs first has same mechanism to early LTP. But prolonged early LTP and induction of LTP can change from transient change to long-term change, and this long-term change need to protein synthesis, and by inhibiting it, we can decrease in maintenance of LTP. Also, a neuron maybe has a neuron can receive different synapses from different neurons. Maybe some of them are silent, and by a stimulation of this neuron, only some of them get activated, because this one is silent. Why it is silent? Because this type of synapse don't have any AMPA receptors on their membrane. We have an NMDA receptor, but we don't have any AMPA receptor, and without AMPA receptors, we can't produce depolarization. For opening of an NMDA receptor and relieving of that magnesium, we need depolarization, and for this depolarization, we need to these AMPA receptors. In silent synapse, this AMPA receptor is internalized, and we don't have any superficial AMPA receptors. But after induction of LTP, because of that phosphorylation by protein kinase C or protein kinase M zeta, we have moving of this AMPA receptor and exposing it towards synaptic cleft. After this LTP induction, also we have responsivity of and on silencing of this receptor. Therefore, in addition to this change, also by this movement of this AMPA receptor, we can and we can unsilent a silent synapse during LTP. Let me to explain it. After that, I will give you rest and I'm in order of the organizer of program. After that, I will continue with LTD. We have different synapses in this figure. This one is weak input. This weak input is not enough to depolarize enough the postsynaptic neuron and remove that magnesium. And because we don't have any opening of NMDA receptor, we don't have any LTP induction. But if you apply a strong stimulation, because of a spatial summation from different input, this postsynaptic neuron reach to threshold level, and we have enough amount of depolarization. And by this depolarization, we can open NMDA receptors, and by opening of them, we can induce LTP. We name this cooperativity. This same to that motivation. In this situation, you don't have any motivation. It is very difficult. You read a book and tomorrow you forget everything. But when you have motivation, and this motivation helps you, and when you have motivation, you concentrate and focus on that topic, and therefore you can add different input to this synapse, and you have cooperative action, and you can induce LTP, and you can remember this for a long time. First, let me to explain about synaptic specificity. In this one, you have a strong stimulation. Each of these stimulation is enough for induction of LTP. When you apply very strong stimulation, you can induce LTP. During LTP, as I said, we have some chemical and protein expression in postsynaptic neuron. All of these synapses are from one neuron. But after induction of LTP, we have synthesis of new protein in cell body. This protein can reach to all synapses, to this stimulated one and to this that is not stimulated. Interestingly, we have induction of LTP only in a stimulated one. Only in a stimulated one. However, we have proteins in this neuron, and this protein can reach also to this unstimulated. Why we have only induction of LTP in this stimulated synapses? Because of tagging. During each stimulation, we have some phosphorylation of some protein in the membrane of postsynaptic spines, dendritic spines. This polarization acts as a tagging. And because this synapse has tagged, therefore, when we have when we synthesize a new protein, this tagged synapse can capture that protein, but this one can't. Therefore, we have synaptic specificity. Only the synapses that are stimulated can induce LTP in that neuron, and other dendritic spine and other synapse of that neuron can't produce LTP, and we don't have no LTP in this one. However, all of them are from one neuron. And associativity. In associativity, if you same to conditioning, Pavlovian conditioning, you accompany one weak, for example, ring, and one strong stimulation, food. Because both stimulation can tag this membrane, after a strong stimulation and induction of LTP, we have synthesis of protein, this protein, in addition to capture by this synapse, because also this one is tagged by a weak stimulation by ring, can induce LTP, but this one not, because of synaptic specificity. After induction of LTP, by removing that food only by ring, you can increase salivation in dog. Pavlovian conditioning. Associativity is one of the mechanisms for this. Now, if you are agreed, I can give you 10-minute rest. After that, I will continue. Thank you.
Hello! In this video, Dr. Tahmasbi, who holds a PhD in physiology and is a member of the scientific board of SBI Institute, will be elaborating on one of the SBI devices called ELAB. ELAB device can record various kinds of extracellular techniques, both invasive and non-invasive signals in vitro and in vivo. ELAB device has the ability to record various kinds of extracellular techniques, both invasive and non-invasive signals, in vitro and in vivo. Field potential, LTP, and synaptic plasticity are some of the techniques used in these kind of recordings. For such experiments, ScienceBeam devices come in great quality in both software and hardware. The signal-to-noise ratio is very high. ELAB is basically all-in-one device that serves all the purposes in the field of electrophysiology studies. ELAB is capable of generating various stimulation pulses. This means you can simply create every stimulation pathway on this software based on your chosen protocol. One of the techniques used for learning and memory study is LTP. For LTP, we can set the current stimulation at prefrontal pathway and record signal from dental gyrus. Here, we want to run an LTP technique. First, prepare the rat brain on stereotaxis device to find the surgical points. Do your measurements based on Paxinus Atlas. Find and mark DJ points and prefrontal pathway. We use stainless steel Teflon coated bipolar electrodes for stimulation and recording. Put the electrodes in the marked points to record. Now we acquire a shape of EPSP. As you see, by changing the electrode's place, the signal's shape respectively changes. Keep changing the setting until you obtain the standard signals. In the meanwhile, you can always refer the acquired shape of EPSP to the standard forms in your references or related articles. eProbe is a powerful and user-friendly software which can change the setting of amplifier easily. As mentioned before, we can create variety of stimulation protocols. For this experiment, we need HFS protocol. Thus, let's run a high-frequency stimulation. Use HFS protocol. For this purpose, we use a current stimulator. Can change its setting in the eProbe software. Keep changing the current until obtaining the desired response. One of the enhanced features in the software is the analyzing option. We have designed a user-friendly and powerful environment for studying any kind of electrophysiological signals such as local field potential, NCV, extracellular multi and single unit recording, electrocardiography, ECG, EMG, and etc.
Sorry, can you see my slide and my voice is clear?
Yes.
Okay, thank you, welcome. Okay, until now, I explained about LTP, line-term potentiation, and its induction in lab, in experimental model by high-frequency stimulation, by tetanization. But in fact, in our hippocampus, barely we use high-frequency stimulation. Rather than in natural way, we have a spike timing dependent plasticity, SDP. In this model, we have presynaptic neuron and postsynaptic neuron. In hippocampus, in natural situation, we have simultaneous activity of both neurons with low frequency. But the presynaptic neuron must just activate before postsynaptic neuron. Therefore, rather than high frequency stimulation, we have simultaneous activity of presynaptic and postsynaptic neuron, and both of them produce action potential, and this firing is coincident. But presynaptic neuron just before postsynaptic neuron, because as I said, the NMDA receptors are blocked by magnesium, and this magnesium must be removed by depolarization. And also we need to glutamate here. Both of them must get depolarized and produce action potential. And when the first, as I said, we have first, our criterion for this type of natural LTP is a firing of presynaptic neuron just before postsynaptic neuron. This glutamate release, and we have glutamate by production of action potential, by producing of action potential in postsynaptic neuron. We have removing of this magnesium, and this glutamate can open this NMDA channel, and we have LTP. But if postsynaptic neuron fire before presynaptic neuron, or we have a firing of action potential more than 100 milliseconds after presynaptic neuron, we don't have any LTP. We have LTD, suppression of LTP. I will explain about LTD. This is the natural mechanism that I want to explain only, but because our concern and our topic is LTP in experimental model, I will explain more about that. But you have to know this is the real mechanism in our hippocampus for induction of LTP. LTP is important for spatial memory. To evaluate this, we have a model for evaluation of spatial memory that is Moore's water maze. In Moore's water maze, we have a pool that is filled with opaque water. And we put a escape platform in the water, but beneath the surface of water, 2 centimeters under water. Therefore, because this water is opaque, this rat can't see this platform. We release the rat from different sides of this pool, and this rat swim and accidentally can find this platform, and because it like to escape from water, sit on it. After that, we repeat this action through different days. You can see by passing days, day by day, the escape latency, or the time that a rat find that escape in that escape platform, decrease by day. Why? Because rat, according to cues that we can attach them on the wall of the room that this pool is located, according to the place of this platform, with respect of these cues, can find. Same to us when you go to your work, according to the shape of a tree, this street, everything, you can find your way. Exactly, we have this situation, this rat, according to some cues, some signs that you attach to on the wall of that room. This rat can find the exact place of platform and can sit on it. And we have decrease in the time during different days. This decreasing manner in escape latency show you learning, spatial learning in Moore's water maze. But in the last day, we remove this escape platform from the pool and we release the animal in the pool. Because the animal remember the exact place of this escape platform, start to swim and it can't find it. Therefore, it starts to search in this quadrant of pool and it spends more of its time in this quadrant rather other quadrant. This shows you a spatial memory. We have a spatial memory in this situation, and this green bar show you a higher amount is spent in target quadrant. Now I want to explain about the relation between long-term potentiation and spatial memory. I said for long-term potentiation, we need 2 activation of NMDA receptor. If you block NMDA receptor or use mutant rats, that this type of rats can't express NMDA receptor. You can see this basic activity and basic response after induction of LTP. In intact rat, we have induction of LTP and maintenance of LTP. But in mutant rat, we don't have any NMDA, we don't have any induction of LTP and maintenance. Also, you have this situation in spatial memory. You can see we have decrease in escape latency in learning phase of spatial memory, but we don't have any decrease or non-significant decrease in mutant rat. Also, when we put the rat and release rat in pool in the last day, you can see he starts to swim randomly in all quadrants, aimlessly swim because he didn't learn and he didn't have any memory about the exact place, and the answer and the results is according only to chance. 20 for each quadrant, we have 4 quadrants and 25% for each quadrant, and the result is only according to chance rather than to memory. Also, if we increase the expression of NMDA receptor, we have increase in LTP induction and maintenance. Also, you can see we have decrease in escape latency, better learning, and increase in quadrant time, better memory in rats that express more number of NMDA receptor that show you the straight relation between spatial memory and long-term potentiation. Now I want to continue with long-term depression, another form of synaptic plasticity. If we have only LTP, we reach a point that which saturate all of the synapse, and because the capacity of each synapse is limited, and you can induce this LTP, induce this chemical and structural change to a limited amount. After that, you can't save any new memory. Also, because we have different dendritic spine from this postsynaptic neuron, some synaptic tract are related to some memory, and some of them are related to another memory. And we have synaptic specificity, and they are separated, separately activated because of that tagging of the proteins, the superficial proteins, because of their phosphorylation that induce LTP only in this stimulated synapse. But if we have only LTP, by time all of these synapses with different types of memory induce LTP and reach to a saturated state, and in this situation we lose our memory specificity and therefore interfering different memory with each other. This is a very bad situation and a weak situation of weak point of LTP. So we need a mechanism to downregulate synaptic function and suppress this LTP. This mechanism is LTD, long-term depression, that prevents from LTP saturation, enhance flexibility in memory storage because of specificity in memory recall, as I explained. But what's the mechanism of LTD? LTD is same to LTP. Both of them can induce by frequent stimulation. But in LTP, you have to apply high frequent stimulation, but in LTD, you have to apply low frequent stimulation. Also, as I explained in spike pairing, as I explained in spike timing dependent plasticity, in spike pairing, in presynaptic and postsynaptic, I said that firing of action potential must occur in presynaptic just before postsynaptic neuron. But if we have firing of action potential in postsynaptic before presynaptic, also we have LTD. I will explain about the exact mechanism. Both LTP and LTD need two NMDA receptors, and also both of them need to calcium. Now the question, how can NMDA receptor play both play a role in LTP and LTD? The key point in this context is the protocol of stimulation. As I said, when you apply high frequent stimulation, you have LTP, but when you apply low frequent stimulation, you have LTD. During high frequent stimulation, because you have higher amount of depolarization, you can open more number of NMDA receptor and more concentration of calcium come into the cell. But in LTD, you have apply low frequency, therefore you have lower amount of depolarization and lower chance of opening of NMDA receptor and lower concentration of calcium. We have two important enzymes in this respect. calcium calmodulin protein dependent kinase 2 or CAM kinase 2. That is important for LTP. I said this enzyme, by phosphorylating AMPA receptor, increase the number of AMPA receptor and activity of AMPA receptor on dendritic spine. But we have another enzyme that is calcium dependent phosphatase or calcineurin. Calcineurin, that is a type of phosphatase, can dephosphorylate AMPA receptor. By dephosphorylating of AMPA receptor, a cell internalize it and drag it toward intercellular space and remove it from synapse, and therefore decrease the excitability of postsynaptic neuron. The affinity of phosphatase for calcium is higher than calcium in CAM kinase 2. When we have high concentration of calcium during LTP, this high concentration of calcium can activate calcium calmodulin kinase 2, but during LTD, because of low concentration and high affinity of this phosphatase, we have binding of calcium to this enzyme and the phosphorylation of AMPA receptor, and by removing AMPA receptor, we can decrease excitability of neuron and we can suppress that LTP. If you remember, I explained in silent and unsilent. During silent situation, we don't have any AMPA receptor. After induction of LTP, we have expression of AMPA receptor on the surface of membrane in synaptic cleft. During LTD, low frequency stimulation, also we have removing of this AMPA receptor from cell membrane. This show you basal activity after low frequency stimulation and induction of LTD. We have decrease in synaptic transmission, and you can see maintenance of this LTD. But if you use a PV that is antagonist of NMDA receptor, you can see this low frequency stimulation is not effective for induction of LTD or maintenance of it. That show you again also LTD obey the Hebb rule. Also, this figure show you this situation LTD in mutant rats, that when you prevent from expression of phosphatase, you can see we can't, in red diagram, we can't induce LTD. But when you off this gene, we have same response that show you LTD, and this phosphatase is very important and functional in induction of LTD. But in Morris-Water maze, during LTP, during learning phase of Morris-Water maze, both mutant rat can't express phosphatase and express LTD. You can see both of them can show they can learn and decrease in escape latency, because we have LTP, and for LTP we don't need to phosphatase. But if you dislocate the escape platform to another quadrant, when you release the rat inside the pool, first it starts to search for this plat in this quadrant. When it can't find it, it starts to search other quadrant to finding that platform. This show you flexibility in that rat, and also show you LTD. This suppression prevent from longer remaining in this quadrant, and we have a shorter time. After that, it learn again for finding this plat in this quadrant. But the mutant rat that can't express phosphatase can't suppress LTP and start its searching to find that platform in this quadrant, and spend more time Eventually, it can learn for finding this new place for plat, because we have CAM kinase 2. Also, we have LTP in normal situation, but this show you that LTD is very important for this flexibility. In addition to NMDA receptor, also NMDA receptor, in addition to its ionotropic action, also has some metabotropic action. And in addition, we have a G protein coupled metabotropic glutamate receptor that through MAP kinase, mitogen activated protein, both of these situation can activate phosphoprotein phosphatase, or through other pathway can decrease the glutamate release from presynaptic neuron, and also remove AMPA receptors from postsynaptic, and by this way can suppress LTP and decrease LTP. And finally, I want to explain about paired pulse facilitation and inhibition. These two methods that you can use in field potential recording are important for evaluation of short-term memory. LTP and LTD are useful for evaluation long-term memory, but paired pulse stimulation is suitable for short-term memory, and also is important for evaluation of the activity of presynaptic neuron, and show you mechanisms that are related to presynaptic neuron. First, let me to explain it simply. You have presynaptic neuron and postsynaptic neuron. In paired pulse stimulation, you apply two consecutive stimulation with same size. Sorry, because I'm drawing it with mouse, it's difficult for me. But these two stimulation, the intensity of these two stimulation is equal to each other. After the first stimulation in second neuron, in postsynaptic neuron, you have EPSP. And after the second stimulation, you have EPSP, sometimes more amplitude, sometimes very small EPSP. Paired pulse facilitation, paired pulse inhibition or depression. Two equal stimulation, intensity of these two stimulation is equal, and you can apply this stimulation consecutively beside each other, and sometimes you have facilitation, sometimes you have depression. Why? The key element and key factor for facilitation or inhibition is interstimulus interval. In dentate gyrus, if you apply two consecutive stimulation with interstimulus interval between 10 to 40, you have depression. But if you more than 50 to 1000, you have facilitation. If you more higher than this value, because they are very far from each other, can't affect on the response of postsynaptic neuron, and we need a limited time to have this situation. If I want to do diagram of response in dentate gyrus, you can see when you have interstimulus interval between 10 to about 40, you have depression. The second response after the second stimulation is smaller than the first one. But after 50, when you have 50 millisecond interstimulus interval, we have facilitation increase. You can see small PS population spike, very large and tall, very high amplitude population spike increase in response of second stimulation. And you can see we have increase in response when you increase the interval between two stimulation, because we don't have any effect it come back to basal activity. Now I want to explain about the exact mechanism. In pair pulse facilitation, when you stimulate the first neuron first time, we have entering of calcium because of action potential and opening of some calcium voltage gated channels. Calcium come into the cell, and this calcium, by affecting vesicles, release neurotransmitter. And we have EPSP in postsynaptic neuron. Removing of this calcium needs time. If you apply another stimulation before removing this residual calcium, by new action potential and opening of calcium channel, we have new calcium. This new calcium add to previous calcium, and this high concentration of calcium can release more concentration of neurotransmitter, and therefore we have a bigger EPSP in postsynaptic neuron. This is pair pulse facilitation, and show you this is result from presynaptic activity and residual calcium. You can see this is a real recording from one of my research. Now, I evaluated the interstimulus interval between 50 to 250 milliseconds. As I said, during first, we have some calcium and EPSP with this size, and because of residual calcium after a stimulation, second stimulation, because we have new calcium add to previous, we have increase in the size of EPSP and increase in EPSP loop, and also increase in population spike amplitude. But if you apply interstimulus interval less than, as I said, 10 to 40, the second response is smaller than previous. The first one is has high amplitude EPSP, but the second one is smaller. Why? Because of recurrent inhibition. When you stimulate the perforant pathway or Schaffer collateral, especially we have this situation in dentate germs. When you stimulate, first you stimulate a granule cell, and we have the first EPSP. Also, we have a stimulation of basket cell or hilar cell. I will explain to in other slide. By a stimulation of these inhibitory interneurons, they release GABA neurotransmitter, and GABA inhibit this granule cell. When you apply the second stimulation, this second stimulation stimulates this granule cell and produce EPSP. But from previous stimulation by this GABAergic interneuron, we have IPSP, and this IPSP can neutralize significant part of this EPSP from this stimulation. Therefore, we have a smaller response in second stimulation. We have feed forward and feed back inhibition. In feed forward, you can see in perforant pathway, by sending signal to granule cell, stimulate it. We have EPSP from first stimulus, and also it stimulates basket cell, and it inhibits granule cell after the first response. It will suppress for a short duration of time, about 10 to 40 milliseconds. During this time, if you apply the second stimulation, because of this inhibition, you don't have facilitation. However, you have a residual calcium, and you can release a high amount of neurotransmitter. But because of IPSP that is result from this inhibition, this IPSP neutralize the stimulation of second stimulus, and we don't have any significant depolarization, and we can't open calcium channel enough, and therefore we don't have new calcium, or if we have, it is very small, and we can release a few amount of glutamate, and our second response is smaller than the first one. And also we have feedback inhibition after the first stimulation of granule cell. Also it sends collateral to basket cell and hilar cell, and also they can inhibit this granule cell only for 10 to 40 milliseconds. After 40 milliseconds, because of residual calcium, we have facilitation, but during this time the second response is smaller than the first. This is my setup in my lab. You can use your stereotypes for dental gyrus. I adjust my stereotypes apparatus both arms vertically, but I found a method for CA1. For CA1, you can apply one of them vertical for stimulation of shopper collateral, and one of them obliquely. This oblique help you to find the pyramidal neurons in CA1 for in a vaster area, and usually by this method more than 90% of my rats give good recording for further experiment. This show you one EPSP from CA1 and paired pulse facilitation with interstimulus interval about 50 milliseconds between the two stimulation. My lecture is finished. If you have any question, please ask me. Thank you for your attention. Sorry for my bad accent and talking very fast. I hope my lecture be helpful for you. If you have any question, you can ask me, and I will be very glad to answer your question. Thank you for your attention.